All right, good afternoon, everyone. I'm Kevin Caldwell, CEO and founder of Ossium Health. I'm going to talk to you a little bit about the company, what we do, with particular emphasis on a clinical study that we're now doing using bone marrow from organ donors to treat patients with acute myeloid and acute lymphoid leukemia. Can everyone hear me, by the way? All right, excellent. Okay, so to kick off, I want to acknowledge that our research was funded in part by grants from the National Institutes of Health. So Ossium's mission is to improve human health through bioengineering. Ultimately, everything we do is in service of that basic goal. The company's scope is very broad, but our original focus, until this day, our single greatest focus, is on using bone marrow from organ donors to improve treatment for blood cancer. So I'm going to open up by talking a little bit about a little bit of background on blood cancer itself, which is often not something that we see organ donors as being a source for a treatment for. I'm then going to move into bone marrow banking at Ossium and the bone marrow transplant study that we're doing. After that, we'll talk a little bit about graft versus host disease. GVHD is one of the principal complications of bone marrow transplants. It's why leukemia patients often die even after successfully going into remission. And then finally, we'll talk about organ transplant tolerance, which is at the cutting edge of some of our research. Okay, so what is bone marrow? Well, formally, it's the spongy substance inside of bones. Within the bone marrow, there are hematopoietic stem cells. The hematopoietic stem cells produce red blood cells, white blood cells, and platelets. And the entire blood and immune system is generated by these hematopoietic stem cells. And so when we do a bone marrow transplant, we're replacing the blood and immune system of the recipient with that of the donor. For the rest of the recipient's life, they'll produce blood and immune cells derived from their bone marrow donor cells. If you were to take a sample of the recipient's blood, the genetic signature you got would match the donor, not the recipient. It is a permanent form of genetic reengineering that's existed for about 70 years. So the malignancies of the immune system that our current clinical study is focused on are ALL and AML. So acute lymphoblastic leukemia means that the lymphocytes have proliferated out of control, and then acute myeloid leukemia means that the myeloids have proliferated out of control. These are two of the most common types of blood cancers. So we're focused on these in part because the clinical impact of successfully improving treatment and accessibility for these is enormous. So a bit of background on stem cell transplants themselves. About 1.5 million stem cell transplants have been done around the world over the last 70 years. In principle, almost any disease for the blood or immune system could be treated with the hematopoietic stem cell transplant. The basic idea is that because you're entirely replacing the blood and immune system of the recipient for that of the donor, a disease that the recipient had in general can be eliminated. So part of the basic idea behind Ossium is that we think these procedures could be used far more broadly than they are. There are a few reasons why they're not used for more patients today. One of which is accessibility. And so the bar on the far left of this waterfall represents the number of patients each year in the United States who go looking for a donor for a bone marrow transplant. And so for these patients, they've already been diagnosed with a blood cancer. They haven't successfully gone into remission from a combination of chemotherapy and radiation alone. Their medical team has determined that a bone marrow transplant is necessary, and they have actually gone in search of a donor. So of those patients, about 4,000 of them are lucky enough to have a relative who's a good match. Typically, this is a sibling who's a perfect match. A sibling has about a 25% chance of being a perfect match. If you've got a healthy sibling who is a perfect match, you're done. There's about 7,000 people who successfully receive a transplant from an unrelated person. So typically, this is a complete stranger who volunteered to have their own HLA type, their information about their own genetics in a global registry. They've matched this donor, and they're willing to have their bone marrow aspirated, which traditionally meant sticking a needle into their ileum and sort of basically sucking out their bone marrow. It's sort of a painful procedure that typically prevents the person from working for several days. And then finally, there's a small number of people, typically fewer than 700, who receive a cord blood transplant. Cord blood transplants generally are much less effective, especially for adults, because the number of hematopoietic stem cells in an infant's cord blood is just very small. So you add all that up, you still have more than 9,000 patients, right? More than 45% of the people who go looking for a donor do not successfully receive a bone marrow transplant. So why not? So the problem is not that there are no theoretical matches for these patients. Almost everyone who goes looking for a donor has at least one person in the global registries who's a potential genetic match for them. There's more than 30 million people in the registries globally. The problem is that these people are not able to actually receive a transplant in time because either the match can't be contacted. Many people will sign up for the registry when they're relatively young, in their 20s or 30s, and then not receive a call or not match anyone for decades. And by the time the registry reaches out, their information has changed and not accessible. They may have contracted a disease in the decades since they initially signed up from the registry that makes them ineligible to donate, or their life circumstances may simply have changed, and something that they were excited about volunteering for when they were in college may no longer make as much sense when they have more obligations. For whatever reason, they ultimately don't provide bone marrow. And so because of that, 45% of the people who go looking for a bone marrow donor after being diagnosed with blood cancer do not receive a transplant. The vast majority of these patients die. So it is this observation, right, this effective shortage of bone marrow, that is the fundamental inspiration behind Ossium's formation. So what do we do about this? Well, I mentioned a few minutes ago that there have been 1.5 million bone marrow transplants around the world over the last 70 years. Virtually all of those transplants, right, were from living donors of bone marrow. So why is this? It's not because organ donor bone marrow isn't viable for transplant. The bone marrow from an organ donor is viable just as the solid organs from those donors are viable. Historically, it wasn't used because, one, you could do a bone marrow transplant from a living person, and at least for many organs you can't plausibly, but also, two, you can't simply recover bone marrow and provide it to someone who is of similar body size and needs a bone marrow transplant. You have to have a very, very close genetic match between the patient and the recipient. If they're not very closely genetically matched, the recipient can receive graft versus host disease. This is the inverse of organ rejection. It basically, instead of the native immune system attacking the foreign organ, the person has a new immune system that attacks every other cell in their body. The disease itself is lethal and can be more dangerous than the leukemia you're trying to cure. And so getting a close match is incredibly important. And the probabilities on that are quite low. If you pick two random people, the odds of them being a match for each other are negligible. And so that means that in order to make this work, in order to recover bone marrow from an organ donor and use it to transplant, you have to have some method, not just of processing the bone marrow, but of cryopreserving it, of banking it, so that one day in the future, when a person who is a match for the recipient comes around, you can provide the bone marrow to them. And that is exactly what we do. So from living donors, the traditional source of bone marrow I mentioned earlier is the ileum. You just aspirate the cells from the hip, basically. It turns out, one of the things that we did at the very beginning is we looked at essentially the entire skeleton. And what we found is that by far the largest concentrations of the hematopoietic cell-rich bone marrow in the adult body are in the vertebral bodies. They're in the spinal column. And so there's lots of obvious reasons why it's not safe to try to tap the spine of a living person for bone marrow. But from an organ donor, it really is an ideal source. And for the most part, before Ossium, most organ procurement organizations were not regularly using vertebral bodies from organ donors. And so we built partnerships with now 25 and counting of the 57 OPOs around the United States to recover vertebral bodies for Ossium. Those VBs are shipped to our facility for processing, tissue and cell processing. We obtain whole bone marrow from the donors. We cryopreserve that bone marrow locally on site in liquid nitrogen vapor. The genetic information about those donors, or at least the HLA type specifically about those donors, is provided to be the match, which is the United States' a bone marrow transplantation registry. They're the people who run the living donor-based bone marrow transplants, and then those cells can be made available to blood cancer patients. And so the basic idea here is to take that number, that almost 10,000 patient with a treatable cancer number who are dying needlessly because they can't get a donor, and slash that. We think we can bring the number of patients who go looking for a donor and fail to receive one down from around 45% of patients to about 5 to 10% of patients within a decade. We want to basically get everyone a bone marrow transplant. That's the idea. So one way of framing this is we're taking all those steps that historically were done after a patient was diagnosed with a hematologic malignancy requiring a bone marrow transplant, finding a donor, contacting them, doing confirmatory HLA typing, scheduling the donor to have additional testing done to confirm that they're still healthy, aspiration of the cells, and finally distribution. We're taking all that and we're doing that up front. And that means that when we have a patient, all we have to do is confirm that the HLA type of the donor matches the patient, and then we can distribute. And so this is going from a process that takes 3 to 4 months to 3 to 5 days. One way of thinking about this is that we're taking a process that really is a service. It's something that is sort of bespoke to the particular patient and that happens only after the need arises, and we're turning it into a product, something that's manufactured in advance and is simply taken off the shelf and delivered. Part of the vision behind this is just dramatically improve the, so I've emphasized so far that there are many people who just don't get a transplant, who we want to get one for. That is a huge part of the goal, but the vision is bigger than that. The part of the idea is that even people who would have gotten a transplant anyway, what we find is that they have much better long-term overall survival outcomes if they receive their transplant within a few days or a few weeks rather than several months. In fact, the probability of a patient still being alive at the end of 5 years declines by about 1% for every 3 additional days that they wait after looking for a donor. And so by dramatically shrinking these timelines, we really can improve outcomes even for patients that would have gotten a transplant anyway. Okay, so what does the study look like? So for this initial study, we call it the PRESERVE study, both because the goal is to preserve human life and because we use cryopreserved bone marrow for it. And we're treating patients ages 18 to 70. Of course, leukemia is a cancer that children do get, but because this is a first-in-human study, we're focusing on adults. We are treating patients with AML and ALL, they're using a conventional sort of conditioning regimen that's relatively similar to what you would get with a living donor transplant. One thing that's innovative about this that I haven't mentioned so far is our dosing. So when you are doing a traditional bone marrow transplant from a living donor, it's one way in which it's similar to an organ transplant is that what you get is what you get. You go in, you aspirate the bone marrow, you infuse everything you get. There's no going, you don't ask the person to go back to the hospital so you can drill into their hip a second time and get more bone marrow for it. You just infuse what you get. And there's not time to characterize the graft, at least not fully before you transplant it. In our case, because the donors because we rely on organ donors and we're using VBs, we're able to recover much more bone marrow from an organ donor than you could ever ethically take from a living donor who still needs their bone marrow. In general, we can get about threefold. It's 3 to 5 times as many cells. And so this has 2 really significant consequences. One is that we can do higher dosing, which is important because higher dosing tends to lead to faster engraftment, which means better survival rates. But more generally than that, we can do optimized dosing. The medical team decides on what dose they want for their patient in advance based on the patient's sort of body weight and other characteristics, and we deliver exactly that for the infusion. Second, it means that in general, we are able to recover enough bone marrow that we can actually do multiple transplants per donor. And so one donor is actually saving multiple lives, treating multiple blood cancer patients through this program. Okay. So there's, let me see here. Okay. So who's doing this with us? Today there's 8 centers that have confirmed participation in the study. Several of these are actively screening patients now. A few others have confirmed through their IRBs that they're going to proceed with the study and are in contracting with us. There's actually about 180 centers throughout the United States that do bone marrow transplants. The top 70 or so of those centers do about 80% of the transplants, and many of those top 70 are participating in this study. And so we really have seen a tremendous sort of swell of enthusiasm from the bone marrow transplant ecosystem. All right. Okay, so how does our organ donor-based bone marrow product compare to living donor bone marrow? Well, we're doing a clinical study that'll be the ultimate answer to that, and perhaps that data will be ready to present at a future event. But for now, what we'll share is our preclinical data. So one of the things that we did early on was compare Ossium's deceased donor-derived bone marrow to living donor aspirate. And if you look at the three charts on the left, this is the percentage of the total nucleated cells that have the CD34 marker. CD34 is just a cell surface protein. It's a marker for hematopoietic stem cells. We look at the viability of those cells, and then we look at their ability to form colonies to grow effectively, essentially. And so the big takeaway from these observations is that there is no statistically sort of meaningful difference in quality between deceased donor and living donor-based bone marrow. And then the graph on the far right, though, tells a very different story. So this is a measure of the T cell count in the products, and what we do see a statistically significant difference in the number of T cells across the two products. There's about a 70% difference in T cells. So that's important because I mentioned earlier that graft versus host disease was one of the most serious complications of bone marrow transplants. That's why a bone marrow transplant can succeed at reconstituting an immune system and eliminating a malignancy and still result in a patient's death because of the rejection that occurs. So that GVHD is mediated by T cells, and naive T cells in particular, and our product has 70% fewer of the cells that drive this complication. And so our hope is that we'll be able to show that patients have a lower risk of GVHD as a result of this. Now, if you put all this together, what we've said is that, one, we can improve access to bone marrow transplants by simply making the cells available readily off the shelf from organ donors so that people don't have to engage in this lengthy search process that many of them don't have enough time to complete. Many of those patients become too weak for a bone marrow transplant before they can finish that 4-month search process. Two, even for patients that would receive a transplant anyway, we're saying we can give them an optimized dose that's specific to their body weight and other needs because we have so many cells per person. And three, we're saying that even those people who would have gotten a transplant earlier will get it faster and have better long-term outcomes. And finally, we're saying even their risk of graft versus host disease is reduced because of the lower T cell count. And so if our goal is to show that there is not some trade-off between increasing the accessibility of a treatment, increasing the affordability of a treatment, and increasing the quality of a treatment, we really are shooting for all three. Okay, so let's talk a little bit more about GVHD. So I mentioned earlier that there's about 20,000 people around the US every year who go in search of a bone marrow donor. That is not the number of blood cancer patients in the US. We have, just with AML and ALL alone, there's about 60,000 people diagnosed with those two cancers combined every year in the United States. Now, so the reason why sort of 40 of those 60,000, the reason why 2/3 of them don't even look for a bone marrow donor is because today the procedure is too dangerous. If it's possible to get your patient into remission from chemotherapy and radiation, there's no reason to risk doing a bone marrow transplant. That's true for the benefit of the patient. Also, even for the donor, asking someone to undergo an aspiration is a significant ask, especially for a stranger. And so our system is designed to try to avoid that, and we really treat the bone marrow transplant as a kind of last resort if other things don't work. But if the procedure were safer, if it were possible to do a bone marrow transplant with a dramatically reduced risk of GVHD, or just if we had a treatment for GVHD that actually worked reliably, then more of those patients could receive bone marrow transplants. Also, the pain and logistical difficulty associated with asking for a living person, particularly a total stranger, to sit for an aspiration is unnecessary if you have a deceased donor source. And so, in addition to the bone marrow product, we've developed a product that's specific for treating graft versus host disease. And so I've spoken largely about the stem cells in the bone marrow so far, but there's a whole gamish of cells that are in the bone marrow. These include mesenchymal stem cells, which have anti-inflammatory properties. They sort of modulate immune function. And we can culture these cells from the bone marrow. We do some engineering on them to sort of accelerate their activation once they enter the body of the recipient. And so before I get into that, I'll just quickly say there's these three different ways of approaching treating GVHD. So acute GVHD is a condition that a person gets within 100 days of their bone marrow transplant. And just remember, conceptually, the disease is leukemia, the treatment is the bone marrow transplant, and then the disease that you can get from the treatment is the graft versus host disease. And so this is a treatment for the treatment. This is a treatment for graft versus host disease, yeah? Okay, so if you, so within 100 days of that, if you get a severe sort of rejection reaction, that's acute GVHD. After 100 days, it's chronic GVHD. This can last for the rest of the person's life. So you could go after either of these, or you could do a prophylactic treatment, which means actually giving a person a dose of a drug before they develop GVHD so that they never do. Okay. So what's the need? Well, the number of people who could potentially benefit from a treatment for graft versus host disease is equal to the number of people who get a bone marrow transplant. And so here you can see a graph of the number of bone marrow transplants that have been done in the US over time. And as you can see that number, particularly focusing on the allogeneic bar, the blue bar, that number has risen steadily over time. And again, there's about 8,000 people who received a transplant successfully in 2019, much lower again than the 20,000 who went to look. Okay. So I'm going to go a little bit quickly through some of these slides, but basically what we find is that these stem cells that we talked about, these MSCs, they actually live in these niches within the vertebral bone. And it turns out that because our OPOs provide us with the actual bone, we're able to get orders of magnitude more of these cells than other groups that are trying to make MSCs, because everyone else just aspirates some bone marrow and they try to grow MSCs from that. We're using the actual bone for the MSCs, and so we get thousands of times more cells than anyone else does. So the basic idea is that because we have so many more cells to begin with because of the donor source, we're able to run our cells through fewer doublings before we dose them, which means that they're closer to the state that they're in the body, they're less manipulated and more potent. Also, we screen thousands of donors every year. Our OPO partners present about 10% of all U.S. organ donors are screened by our donor team, and we really choose, we really optimally choose the best donors for this GVHD product, and we're able to make thousands of doses, really tens of thousands of doses per donor. Okay, and so this program is under IND with the FDA. We have an orphan drug designation for it. The FDA has determined this program is important and valuable enough that they've given us this accelerated approval pathway. And for this study, and this is a separate study from the leukemia study I mentioned, for this study, we are doing a phase 1 with 24 patients. Okay. How am I doing on time?
You still have 5 minutes.
Okay, great. 5 minutes. That's plenty. That's plenty. All right. Okay. So now, so we've talked about 2 programs so far, using bone marrow transplants for leukemia, graft versus host disease for patients who've had bone marrow transplants. I now want to speak about something that's much closer to the sort of daily operating lives of our partners here, and that's transplant tolerance. So, standard of care for decades has been someone gets an organ transplant, they go on immunosuppression. They're on immunosuppression for the rest of their lives, generally. They have an increased risk of infection, increased risk of cancer because of the immunosuppression drugs, but they are able to they're able to take the organ. So this paradigm, immunosuppression has been incredible for the organ transplantation ecosystem. It's enabled over the decades millions of lives worldwide to be saved. But a paradigm where if you get an organ transplant, you have to have a weakened immune system for the rest of your life as a paradigm in which we would argue that the organ transplant isn't actually a cure for organ failure, it's a treatment. It's allowing you to continue to live, but not at the level, at the quality of life that you had before you had organ failure at all. And so our basic idea is to eliminate the need for immunosuppression. So how do we do that? So I mentioned earlier that if we give someone a bone marrow transplant, you are replacing the immune system of the recipient with that of the donor. And so in the context in which I've been talking about this so far, the patient has a malignancy. Their native immune system is cancerous, and so your goal is to completely eliminate it and replace it. In the case of these organ recipients, there's nothing at all wrong with their immune system other than that it doesn't happen to agree with this new organ that they got. And so what we do instead of a full bone marrow transplant is what we call a procedure called immune tolerance induction, where we don't actually ablate their native immune system. They have virtually all those cells are intact. We do, however, give them a dose of bone marrow from their organ donor, from the same person they got the organ from. What we can do is induce a state called hematopoietic mixed chimerism, right? A chimera being an organism that expresses or has cells from two distinct organisms within itself. So in this case, what we can do with this protocol is we can induce a state where the patient long-term is producing blood and immune cells from their native bone marrow and their organ donor's bone marrow. And what we find is that this chimeric immune system can accept cells from the organ donor itself. In other words, you can win them off of immunosuppression entirely. And this isn't just theoretical. We've done this in the clinic. Okay, so part of the beauty of this is that we are banking bone marrow from organ donors every day anyway. That's what we do for blood cancer treatment. I mentioned to you earlier that we recover enough bone marrow from those donors that we don't need to use all the bone marrow from a given donor for blood cancer treatment. The sheer volume of cells we get is much larger. That means that there are cells that are available to use for organ transplant recipients. And so in this case, the bone marrow follows the organ. There's extensive work on this, and extensive published work even, from people who began doing this kind of work with people who received kidney transplants from living donors. This work dates back to the late 80s and 90s. The basic idea was bone marrow transplants from living donors were well established. Even then, organ transplants, kidney transplants from living donors were established. They would just match up the donor, and they would get kidney and bone marrow from the same person. So that's great. But 80% of all US persons who receive an organ transplant, 80% of organ transplants are from deceased donors. And so if we want to bring the clinical benefits of immune tolerance induction to 80% of organ recipients, you have to have a way to recover and infuse bone marrow from organ donors, and you need a protocol that works for that. And so we began with this multivisceral transplant study. Multivisceral transplants are very rare. Many of you have probably never been involved in one, but the reason why they're rare is not because there's not a significant need for it. The biggest reason is just that the transplants don't work that well. They're usually rejected. They're rejected about half the time, and so most of the time the surgeons don't bother with them. The one benefit of the fact that the standard of care is so poor is that if you have an intervention that improves the standard of care, we can get a lot of signal on whether or not we've improved outcome for patients with a relatively small study. If we do 3 to 5 patients and they don't reject, we know we've accomplished, we know the intervention has been effective. And so here we're doing these combined intestine, liver, pancreas, and stomach transplants, followed by a bone marrow transplant 11 days after the organ transplant. We check for T-cell chimerism at 4% or better, and then multi-lineage chimerism at 1% or better, followed for a year. and then we check for graft survival for up to 3 years. Okay, so there's lots of centers that have done and are participating in this, and I'm probably over my time allocation, so I'm not going to go into much detail about those, but it's an active program. Okay, I guess the other thing I would mention is that we've had tremendous success working with the U.S. Food and Drug Administration across the board on improving access to bone marrow transplants, on doing tolerized organ transplants, on treating graft versus host disease. Not only have they, approved a number of our clinical trials to proceed, they've also given us sort of regulatory designations that are encouraging, and we are incredibly excited about completing clinical development and bringing these therapeutics to the market. And finally, we just want to, everything we do is only possible because of our relationships with sort of the organ procurement community and ecosystem. The OPOs have been incredibly supportive. The transplant centers have been incredibly supportive to enable sort of this work and this research. And we want to thank that community. And in particular, I want to acknowledge Nevada Donor Network, which was one of Ossium's very first OPO partners, and really sort of paved the way for a lot of this work. All right, thank you.
Thank you. Any questions?
Well, maybe I lost the moment that you explained at what moment of organ donation actually you obtain the bone marrow.
Okay, so
I have to say that we studied that a long time ago, and it was we did not find a way to do it. So I'm curious to know how you manage that.
Yeah. So the vertebral bodies are recovered within 8 hours of cross-clamp. And so typically what the teams do is immediately after the solid organs are recovered, while the chest cavity is still open, they go in and they'll recover the VBs. They recover the spinal column. That is wrapped, put on wood ice, and shipped to our facility for processing. So one of the things that we did early on is we published a study looking at the relationship between ischemia time, warm and cold, and the sort of the viability of the cells that you got. And what we found is that there was a sharp drop off after 8 hours. If you wail longer than that, then you're not going to get enough cells to do a transplant. And in fact, every hour you shave off of even 8 is significantly beneficial. Not only does waiting longer sort of decrease the viability that you get, it also increases the risk that microorganisms grow and you get sterility challenges. And so a speedy recovery is very important. And actually, for the first several years of our existence, one of the major things that we focused on was optimizing that recovery process, because it is so tremendously important. It's not easy.
Any other questions?
Congrats for your presentation.
Thanks.
Statistically, you need the same number of deceased donors, then compare the living donors to find a match. Yes?
No. So if you were to use the same clinical protocol, you absolutely would. But for our deceased donor study, we're using a mismatched unrelated donor protocol. It allows you to have up to 4 mismatches for the transplant. We use post-transplant cyclophosphamide, and that enables you it reduces GVHD risk and enables you to do a transplant with mismatches. And so you can have a dramatically smaller bank and still successfully do the transplants.
And did you comment regarding the interferon, the injection of interferon? What's the moment you inject interferon in the donor? Do you use the interferon in the donor?
Okay, so I think you're referring to the use of interferon gamma for the mesenchymal stem cell product. So we don't use that for the bone marrow transplants. That's used in the GVHD treatment. And the interferon gamma it's not delivered directly to the recipient. It's used to stimulate the cells, the MSCs, before cryopreservation. So there's several different products in a lot of studies that I went over quickly. Yeah.
And the last question. When you have a living donor, you need to guarantee the traceability and the complete health of the donor, okay, to use the cells, the bone marrow.
Yep.
But how do you do to guarantee the same when I have a deceased donor. Because in deceased donor, you have, for example, a DBD, you have a patient, and it's a different scenario. It's a big inflammatory process. Sometimes you have infection, et cetera, et cetera, et cetera. Use drugs, et cetera, et cetera. How do you do to guarantee the quality of this tissue and guarantee the traceability? Thank you.
So I lost some of that, but I think the essence of your question is, how can we be certain of the safety of a deceased donor-derived bone marrow product, and what are the differences between how we would evaluate a deceased donor bone marrow versus living donor? That's the root of your question? Yes? Okay, so I'm glad you asked that. So the first thing to remember is that all of our donors are qualified solid organ donors. And so if they weren't healthy enough to be an organ donor, we would never see them. They wouldn't be considered at all. We then add an additional layer of screens on top of the requirements to be an organ donor that's based around the requirements for being a living bone marrow donor. And so there's a number of sort of blood diseases in particular that are rule-outs for bone marrow donors that are not rule-outs for organ donors. And so we do additional testing and we apply all of that. We also do sort of product-based quality testing on the final product even after the product is completed. And the products are initially placed essentially in these sort of quarantined, isolated, sort of -80 freezers before being put into final cryopreservation with the cleared products. We review all of the organ charts. We also review all the testing data that we do with our quality team before those donors are sent to our medical director for clearance. And so we actually, in many ways, if we compare the information that we have about our patients to the information you get from a living bone marrow donor, we would argue that we actually have more data. We have all the information that you get for the organ transplant and all the information about the bone marrow and the cells from our additional testing. And our donors don't change. And so there's no waiting period of between when you would test the living donor and then when you would ultimately do the infusion. The donor is cryopreserved. And so we've compared, so I mentioned that we're working with the Living Donor Registry, Be the Match. We've compared the data that we have available on our donors to what they get on living donors, and there's no fields and there are no questions that you might ask about a living donor that we're not able to answer here.